Dr. Umayal Ramanathan College for Women, Karaikudi, Tamil Nadu. Today's topic is Today's topic is design, drug discovery, and the elucidation of mechanisms of biologically active components. Ma'am, this time is yours.
Yeah. Am I audible to all of you?
Yes, ma'am.
Yes, ma'am.
Thank you so much. If there is any hindrance or any connectivity issues, you can please let me know so that I'll continue from that slide. Okay. So let's
Yes, ma'am.
start.
Is it audible, ma'am?
Yes. Thank you so much, Rekha. So the slides are visible, right?
Yes, ma'am.
Yes, ma'am.
It is
Thank you.
visible.
Thank you. Shall I start with the session?
Yes, ma'am.
Yeah, thank you. Good evening, all. Myself, Dr.R.Beema Shafreen, and today's faculty development program, I'm going to deal with the topic that is design, drug discovery, and elucidation of mechanism of biologically active compounds. It is being categorized into three different areas, that is designing a drug, drug discovery and development, and elucidating their mechanism of action. So first we need to understand what is a drug. So drug is a substance when it is intaken by the human system, they get dissolved and they are being absorbed to the different organs, and the active metabolites are being received by the target receptors, whereby there will be a clinical response or a response in the human system. So drug is generally being used to treat illnesses or conditions in human as well as plants as well as in animals. So there are different sources of drugs from natural products. Generally it will be plants, microorganisms, marine organisms. From plants, the major examples are morphine, quinine and aspirin. And from microorganism, well known is the penicillin. It is used as an antibiotic, and from microorganism, it is a cytabrin, which is derived from sequins. Similarly, we have traditional medicine. In traditional medicine system, traditional Chinese medicine TCM and Ayurveda source of medicinal compounds are being widely used for centuries in order to treat variety of diseases. And many modern drugs which are derived from the plants as well as the natural substances are being used for traditional medicine practices. From synthetic chemistry, the chemical synthesis of the compounds, the examples are aspirin and ibuprofen, which is used for treatment of several pains as well as inflammations. And from combinatorial chemistry, the libraries of compounds are being screened for their biological activity, which is leading to the discovery of new drug candidates. From semi-synthetic compounds, generally these are the drugs which are derived from natural sources where their chemical modification is undergone. This chemical modification will enhance their potency as well as reduce their effects. Such examples are hydrocodone and oxycodone, which are derived from the morphine structure. The other biotechnological methods which are used as a source for drug is recombinant DNA technology, well known as insulin, and the monoclonal antibodies which are used in cancer therapy. In gene therapy technique, we have delivery of genetic materials into the cells which can treat as well as prevent the diseases. Another highlighted technique is the virtual screening and drug designing, where the drug molecules are being screened using computational models and also through simulation methods. We use large libraries of data sets in order to screen for the chemical compounds and predict their biological activity against the specific targets. So the key concept for drug designing is to find new medications that can effectively target the biological process or the molecules that are involved in the disease. The first concept will be the understanding of the biological targets. Here the targets will be proteins, enzymes, receptors and nucleic acids. And their molecular interaction needs to be studied, where the protein and the receptor will have interaction with the ligand molecules. So the binding effect can be studied, whether the binding can lead to the blocking of the activity or modulation of its function, which can achieve with the desired therapeutic effects. Next, the shape and the charge complementarity needs to be studied, whether it is fitting exactly into the binding site, like a lock and key, and it is forming stable conformations or leading to the desired biological response. The computer modeling and bioinformatics is a key concept where you have a large data set of compounds, where you predict your molecular interactions, optimize the drug candidates, understand their structure activity relationship, and pharmacokinetic properties of the potential drugs. Next, the key concept for the drug designing will be optimization and development, where the chemical properties will be improved in order to their potency, selectivity, solubility, and stability. Thereby, their toxicity can be minimized. The other biological therapy is the therapeutic antibiotics, which are being produced by engineering the proteins. Protein-based drugs are being enhanced or being designed in order to enhance the affinity and selectivity to the and stability of the particular protein to the particular target of the disease-causing agent. So next concept will be the clinical translation, which has been mainly focused, where the scientists will come up with a new idea, where the compounds which are being identified will be taken for testing in experiments, as well as in vitro experiments, as well as the in vivo animal studies. And finally, the successful compounds will be taken for the clinical trials, and it will be approved by the regulatory agencies, and the approved drugs will be with the safe and effective efficacy will be taken by the doctors to be used for treatments to help the patients. The regulatory approval and the market availability is also one of the major key concept of drug designing, where it needs to demonstrate the safety and efficacy in clinical trials that has to be submitted to the regulatory approvals from the health authorities, and they can be manufactured and made available for the patients for clinical use. So the drug design process for each and every disease is, first, for the cancer treatment, we have to identify the protein which are involved in the overreactive, that is the overreactive of the cancer cells can lead to the prevent to the growth of the cancer cells. So the protein, small molecules needs to be designed for the target protein, and they have to be fit into the active site of this protein. Thereby they can block it, and the function can be stopped, and thereby the cancer cell growth can be stopped or reduced. And the next one is the antibiotic development, where the structure of the bacterial enzymes will be studied, which is involved in the cell wall synthesis. The molecules that are going to mimic the shape and the charge and the size of the particular enzyme substrate will be designed, and that particular region will be designed and targeted by that small molecule which can bind to the enzyme, and thereby they can inhibit the bacterial cell wall formation and lead to the killing of the bacteria. In Alzheimer's disease, we can identify the abnormal protein aggregates which can form amyloid plaques in the brains of the Alzheimer's patient, and molecules needs to be designed against these amyloid plaques, and we have to check whether it has to penetrate the blood-brain barrier, cross the blood-brain barrier, and then bind to the plaques. If they are binding, they can prevent formation of the plaques or promote their clearance and potentially slow down the progression of the disease. In vaccine development, vaccines can be designed by identifying the antigenic epitopes on the pathogens, then peptides or the recombinant proteins that are mimicking these epitopes can be designed. This can lead to the stimulation of the immune system to produce protective antibodies against these pathogens, leading to the development of vaccines. In biological therapies, you have computational methods where you engineer the antibodies for improved affinity, selectivity, and stability, modify the antibody structure, and enhance its ability to target the specific disease-causing molecules, thereby minimizing its side effects. So the overall drug discovery process is addressing the unmet medical needs, improving healthcare consequences worldwide, for which we have to understand the disease burden. So most of the well-known diseases are cancer, infectious disease, cardiovascular disease, where the neurological disease which are leading to the morbidity and mortality rate is high. So the effective treatments are required, thereby the elevating suffering can be reduced, and the mortality rates can also be reduced. So the key solution for this is to identify novel therapeutic agents that can target the specific disease mechanism or the pathways, develop drugs that are safer, more efficient, and have smaller side effects, and can be a replacement for the existing treatments. So the science streams that are working in intersection is structure biology, biochemistry, medicinal chemistry, toxicology, pharmacology, biophysical chemistry, and informational technology, which are breaking through therapies that can transform patients' care and extend life expectancy. So personalized medicine is also playing a major role here in drug discovery, which enables the development of tailored medicines to the individual's patients that are being characterized. So here it will be involving the therapies based on genetic, molecular, and other biomarker strategies in order to optimize the treatment. So economic impact of drug discovery is pharmaceutical industry have a very good contributed to a significantly economic growth, and variety of job creations are being observed, and the successful development has led to the commercialization of innovative products and generate revenue and fostering investment in the research and the development sectors. So the drug discovery is addressing emerging challenges such as tackling the emerging health challenges such as antimicrobial resistance, rare diseases, and global pandemics, and thereby it has enabled rapid response to public health crisis by developing vaccines, antiviral drugs, and other interventions. So the collaborative effect required is from academia, industries, government agencies, and non-profit organizations, and it is also fostering knowledge sharing, resource pooling, and cross-disciplinary innovations are being studied. So the statistics of drug discovery is it is a new tool to fight sickness, where the duration is about 13 to 15 years. In early discovery, it starts with the screening of potentially active compounds. Then the compound testing will be performed for the safety and the effectiveness, and the successive rate will be out of 5,000 drugs, only one will be taken into the market approval. And for preclinical testing, out of the 5,000 to 10,000 drugs, 250 will be taken for the candidate for the next stage. So the cost to the research and develop a new drug is generally estimated as $2.6 billion, and the post-marketing and the monitoring and development cost is around $312 million. The total life cycle for research and development of this new drug is approximately estimated as $3 billion per drug. So this is the timeline image for drug discovery, where the first stage of drug discovery is we have thousands of compounds, and the second stage will be the screening out from 1,000 to 250 compounds. It is called as the preclinical development. This is an overall, it is 3 to 5 years. This takes around 3 to 5 years. A thesis work can be around 3 to 5 years. We all stop our thesis work at this stage, and we did not extend it to the next level. So out of 250 compounds, we have to cut it down to 5. Where this part requires stage 3, that is the clinical development, requires more money, and it is taking around 5 to 7 years for it. And then it goes for the regulatory approval, which is around 2 to 3 years, and finally we come out with one successful compound or the lead, which is going to be the drug molecule. So the steps in drug development is the basic research. We have number of compounds here, that is around 10,000 compounds. Then we have drug discovery, 5,000 to 20,000 compounds. Then the preclinical research, we have 250 compounds cutting down. Then clinical research, five compounds are getting down and cutting down. And finally, we have an approved drug. So we have 4 stages for drug discovery process. The stage 1 is the early drug discovery. So this stage, which we all carry out in our laboratory, we easily carry out in our laboratory. That is, first we screen a large number of targets from the disease-causing agent. Say, for example, if it is a bacteria, this bacteria is involved in a type of disease, cardiovascular disease, or it is going to cause streptococcus pyogenes, it is going to cause rheumatic fever. In that, from that organism, we are going to take out the proteins, and those proteins will be the targets, and these targets will be used for screening for large libraries of chemical compounds. This process we call it as high throughput screening, that is HTS, or high content screening, that is HCS. So once the compounds are being screened, we will be supposed to see whether they are interacting with the target protein, and so some show some potential therapeutic effects. These compounds, we cut it down and showing the promising effect, we call it as hits. This hits will be later on taken to the next step. It is called as the development of assays, where we do different activities for the compounds in order to further screen and perform the prioritized most performing lead compounds will be identified. So hit to lead. So this is the phase we get with hit to lead, and then this lead will be generated for testing for their pharmacological activities, like their potency and their selectivity. What are all the properties that could lead them to an suitable drug moiety. So this properties will be improved at this stage, and then they enter into a state called as lead optimization, where we undergo iterative optimization through medicinal chemistry in order to improve the drug-like properties. And finally, we'll be going for in vitro and in vivo assays, and where the evaluated compounds in the laboratory will be tested in animal models in order to assess their efficacy, pharmacokinetics, as well as toxicity. Stage 2 is the preclinical phase, where the refinement and the optimization will be carried out. In refined and it will be measured for their safety as well as efficacy. Then it will be taken for laboratory testing, where the pharmacological properties will be tested, and animal testing will be carried out. In animal testing, it will be more on the pharmacokinetics effect, and finally it will be taken for production in order to scale up. This production for scaling up of the product will lead to the compounds for further clinical studies. Stage 3 is the clinical phase. This phase is actually classified into four. The phase 1 will be starting with a small scale study, and the healthy volunteers will be involved. In the healthy volunteer, subjects will be around 20 to 80 individuals will be involved. And this phase will mainly answer the crucial questions about the safety profile as well as the potential side effects. The questions will be like, is the drug safe? What dose can be given without side effects, and how does the substance behave in the body? So these main questions will be addressed at this phase. And then before conducting the phase trials, the active ingredients will be manufactured using GMP conditions in order to ensure their quality and consistency for the next clinical process of testing. So the phase 2 of this clinical phase will be large-scale studies in patients, where a group of adults, that is 100 to 500 patients, will be taken. So previously it was on healthy volunteers, and here it will be the patients, where they will be given a dosage in order to check the effectiveness, tolerability, as well as the dosage of the drug in large groups of patients. And primarily it will focus on confirming the therapy concept, which is going to be the proof of the concept. So it is this phase itself is further classified into 2A and 2B, and 2A is confirming the therapy, and 2B is to determine the appropriate dosage. So then you have phase 3 in clinical trials. This phase 3 is the large scale studies conducted on patients. Previously it was small scale in patients, and this is large scale in patients. Similarly, here also the drugs will be given to the patients, and it will be compared with the standards and the controls. Control and the standard would be compared for the confirming their efficacy, and their monitoring of their safety, how far, whether there is any adverse effects or side effects associated with the drug candidate, and also with the standard and the control, it will be evaluated whether the drug is acting superiorly or non-inferiorly or in equivalence. So the design is in such a way that randomized control trials will be taken, and large sample size will be involved, and treatment groups will be involved. So in randomized control trials, it will be like assigned to a treatment groups to minimize the bias, and it will also allow for valid comparisons. Whereas in large sample size involves thousands of patients which are being recruited from multiple clinical sites in order to increase the statistical power. And similarly, in treatment groups, the patients may receive the drug candidate and the standard treatment or the control depending on the study protocol. This will be tested. And finally, the confirmation of efficacy, it has a primary endpoint and secondary endpoint. In primary endpoint, the disease progression, survival rate, and symptoms of improvement will be studied here. And in secondary endpoint, it will be including the quality of the life, symptoms of relief from that particular disease, and the biomarker levels will be studied at this secondary endpoint. Finally, a statistical analysis will be performed, and this data is very useful for the phase 4. So safety monitoring, in that after doing this confirmation efficacy, then we have to move on to the safety monitoring. In safety monitoring, it will be having a safety profile where the drug candidate, including any severity in the adverse events, will be noted down, and the risk benefit assessment will be carried out, where a balance of potential benefits of the treatment with the risks of adverse events to determine the overall safety will be calculated. And then comparison of the standard and the control will be carried out. The standard here will be the comparator groups, and the superiority, inferiority, and equivalence group will be there. Then you have clinical relevance. In comparator group, the efficacy and safety of the drug candidate to the standard treatments or the controls will be carried out. And whether the drug is superior, inferior, or equivalent to the other existing therapies will be evaluated and recorded. And in clinical relevance, the treatment effects will be noted down. So overall, the phase 3 is playing a crucial role in providing evidence needed for the regulatory approval. Once all the data is being collected, it is going to give an efficacy and monitoring the safety, and it is also giving a compared standard data, which is essential for healthcare decisions and in order to improve the patient's care. So once the data is collected, it will be going on to the next phase, that is the phase 4. Phase 4 is the post-marketing surveillance studies, which is used to monitor long-term safety and effectiveness of the drug after it has been approved for sale. At this phase, it is approved, and then your major objectives of this phase is long-term safety, real-world effectiveness, and quality of life. In long-term safety, it deals with adverse reactions not observed in preclinical market trials. So previously, whatever the effects are not being noted will be noted at this phase. Then the real world effectiveness, where how well the drug is performing after it is coming to the market, whether the patients in the population or the clinical settings having any effects or any adverse effects will be noted down. Then the quality of the life will be noted down, or it will be reported how the outcomes are, whether there is any improvement or whether the patient is having any side effects. All the qualities will be noted down or recorded down. So these are the major objectives of phase 4, and the study is designed with observational studies, large patient population, and data collection. In observational studies, it is rather than controlled clinical trials. In large patient population, it will involve diverse groups of people in the real world, and they will be noted down for the when the drug is being used for those patients. Then once the data is collected and the patient's outcomes are being recorded, and the healthcare utilization, and will be over an extended period, and then will be given for monitoring the safety assessment, that is the signal detection as well as the risk assessment. It will be systematically carried out in the procedure, where it will be noted down at every time points will be recorded, and it will be noted down about this drug. And this will be a stage called as pharmacovigilance method, where it will be detected for the signals of the potential safety concerns. So after it is being assessed over the risk benefits profile, and the data is then be given for the phase 4 trials. After assessing the effectiveness and the real world outcomes, subgroup analysis, and comparative effectiveness, it will be entering into the regulatory compliances. So regulatory compliances, where the regulatory agencies will be sitting together, and they will discuss about the drugs approval, final approval, and the safety reporting will be given by the regulatory authorities for the ongoing monitoring of the safety effectiveness. And finally, the drug will be approved by them, and it will be taken for the market to access. So the importance of this phase 4 trial is critical, and it is going to be a pharmacovigilance effort. So we all know now, and I hope you all understood about this pharmacovigilance effort. It is just grouping all over the world and studying about the drug, how it is being affected. Then the safe and effective use of the medications in the clinical practice will be studied, and there will be continuous monitoring in the real-time use, during the real-time use, and the patient safety will also be recorded in order to improve the healthcare outcomes. So the final stage is your regulatory approval. This regulatory approval, The data which is collected from the clinical trials will be submitted or compiled into a new drug application or biologics license application, and it will be submitted to the regulatory authorities, and the regulatory authorities or FDA and EMA, they will review and approve our application, and finally the regulatory review will evaluate the data to ensure the candidate drug is safe, effective, and of high quality. And for approval and marketing, the drug will meet the regulatory requirements for sale and marketing, and the post-marketing surveillance will still continue in order to monitor the drug safety and effectiveness in the real-world clinical settings. So this is the overall drug discovery step where you have the therapeutic targets being identified, leads will be discovered, leads will be optimized, then the clinical candidates will be studied. So this too comes under drug designing, and finally after the clinical candidate will enter as a commercial drug. So how the lead is being discovered? So lead is being discovered through different methods. That is, you have the experimental data. The experimental data is through X-ray crystallography or NMR. You have the crystal structures here. This crystal structure, if it is absent, we go for a different process called as homology modeling, where you use the template structure of this model and you can build a model. And if you have only the sequence of this novel therapeutic targets, you can go for comparative modeling with this experimental model, and we can design a 3D structure for the therapeutic target. Once this 3D structure for the therapeutic target is being designed, we use virtual libraries of compounds or chemical compounds, which will be used for high throughput docking studies. From the high throughput docking studies, we will obtain the hit. Or this 3D structure will be directly going for structure-based library design, and the hits can also be obtained by screening the combinatorial libraries. And finally, the hits can enter the structure-based analog designing, where protein 3D ligands will be simulated using a computer, and then finally the lead molecules will be identified. So this is how lead is being discovered using computational techniques. So the main computational techniques used in drug development is target identification. You have bioinformatics. In bioinformatics, you can use sequence-based approaches as well as structure-based approaches. Then you have inverse docking and inverse QSAR. Then you have target validation. You have cavity searching and binding site prediction. Lead discovery, it includes docking, molecular dynamics simulation, library screening, and de novo design. In lead optimization, you have QSAR, pharmacophore modeling, and in vitro ADME. And in drug development, you have preclinical tests, that is phase 1, phase 2, phase 3. These three are clinical trials, and the phase 4 will be your pharmacovigilance. So this is one of the representation of the basic bioinformatics approach for drug discovery, where you have library of compounds. These compounds will be screened using the drug targets, using a computational approach where molecular docking, simulation, or virtual screening can be performed using a computer. And the binding, this is your target to which the receptor to which your ligand molecules are binding, exactly binding to the active site. Then these lead molecules, now called bit molecules, are now called as leads, will be taken for optimization. And further, they are binding after identifying the binding region or the binding site. Then they are being undergone a simulation step, that is molecular dynamic simulation, where energy calculations will be carried out, and finally the lead molecule will be taken for in vitro testing. Using various assays, it will be screened in our laboratory condition, and then it will be taken for in vivo testing using animal models. It could be any type of animal models, whichever we prefer, and then we can go for clinical trials. This is one of the basic approach using bioinformatics. We start with a library of compounds and we end up with an clinical trials. So elucidation of mechanism of biologically active compounds, it is very, very important because it will help us to understand the drug discovery process, how the molecular targets are evolving, and how they are being involved in drug designing, how the targets are involved in this particular activity, or the sites which are being involved. And but it was also playing a major role in personalizing a particular medicine, and it is also it is like we can tailor particular individuals for a drug for an individual patient's um, disease. So the methods for mechanism elucidation is structure biology. In structure biology, we have this X-ray crystallography and cryo-electron microscope, which will reveal the three-dimensional structure of the particular disease drug target, where also the drug target complexes. And using molecular docking approach, you have computational modeling, which will reveal the binding affinity as well as the binding mode of that particular compound into the target site. And functional assays such as biochemical assays, cellular assays, and which will reveal the biological activity, functional effects of the compounds and the target proteins on their pathways. So these are the few case studies which we are going to discuss. First case study will be like inhibition of enzyme activity. In inhibition of enzyme activity, we can demonstrate how one compound blocks the enzyme activity of its target protein. Say, for example, if a particular protein is involved in an fatty acid synthesis, that protein can be targeted, and thereby, if it is targeted, the synthesis cannot be completed, and that can inhibit the complete cycle of that particular fatty acid pathway, and thereby they can either kill the organism, or the production of that particular secondary metabolite can be stopped. So these are the one case study which can lead to either to the downstream effects on the cellular signaling pathways, or it can upstream the process. So inhibition of an enzyme activity is the first case, and second case is like modulation of protein-protein interaction. Modulation of protein-protein interaction is like a compound can either disrupt or enhance the specific protein interactions by influencing their cellular processes. The process can be either inhibited or enhanced. In case 3, the activation of the signaling pathway. In activation of the signaling pathway includes how the compound activates the particular signaling pathway to exert therapeutic effects in the disease state. So this can be also being studied. So these are the different cases for elucidating the mechanism of an action of a particular. These are few examples. Based on your study, it could be modified. Accordingly, you can modify it. So these are the few case studies which I used for my PhD work. So this is one example. You can see that from isolating a natural compound from a particular microorganism or from an environment. Say, for example, a seaweed. From seaweed and bacteria is being isolated, and that bacterial extract is showing antimicrobial activity. Antimicrobial activity can be due to inhibition of the cell wall synthesis, or it could be the inhibition of an protein synthesis, or it could be synthesis of inhibition of the plasma membrane. So either of the other process can inhibit the growth of the particular organism. It can lead to the death, or it can make the organism bacteriostatic. So these are the different mechanisms can be happening. So we don't know at the time point what has happened, but these could be the mechanisms. These are the mechanisms for which the target proteins needs to be chosen from each and every aspect, and the target proteins needs to be screened against the active compounds which has been identified. For that, active compounds needs to be through LCMS or GCMS, the active compounds needs to be identified. After the active compounds are being extracted, and it needs to be targeted into that particular protein through bioinformatics studies, and the active regions where this binding can be revealed. So here the extraction and identification of the bioactive compounds, where they can be scaled up, and optimization and production can be scaled up, and finally when it is scaled up, it can be taken for further in vivo evaluations and clinical trials. So the lead from here, the lead can be obtained, and finally it can be optimized, and the production can be increased and taken for further studies. So this is one of the case study. Next case study is you have an elucidating the mode of action of the compound through the gene expression signatures. So you have a query compound, and you have a cell line. In cell line, you can see that the level of genes are being upregulated or downregulated. If it is upregulated, it could be biological activated pathway and biologically inactivated pathway. So this can be discriminated and they can be produced. And similarly, Prediction of potential target proteins. You have an query compound, and this query compound is used in a particular cell line where upregulation, downregulation, and this query compound binding to the target proteins can be identified. This target protein, on the other end, it is showing the interactome, and this interactome can be involved in a particular disease. Other associated disease can also be studied. And similarly, the similar signature compounds can be identified for this query compound, and that can also be taken to the cell line and can be tested based on the upregulation, downregulation. The proteins that are involved in interaction can be predicted, and their interactomes can be ruled out, and finally involved in the disease can be studied, or it is involved in the disease. Indications of the disease can also be studied. And the next thing will be prediction of new therapeutic indications. So on the overall, you can see predicted associations which can lead to a disease indication can be taken. Maybe this could have downregulated. Because of the downregulation, this disease could have happened. So like this, we can come to the conclusion of why this disease is occurring and how this particular compound is going to play a major role in treating it or upregulating it. And the next case study is activation of the signaling pathways. We have multiple natural compounds from which the active compounds can be taken, and they can be screened for their ADME, that is absorption, distribution, metabolism, and excretion. And this active compounds can be taken for target fishing, and the potential targets can be identified. So once the potential targets are being identified, the pathway analysis can be performed and disease analysis can be performed, and the network constructions can be performed based on the overlapping of these two networks of proteins. So this is how the activation or the downregulation of the signaling pathways can be studied using the compounds which are being used. Next is the overall summary for this particular topic is the drug. In the drug development process, you first select your target, then you go for lead discovery, then you go for medicinal chemistry, and then you go for in vitro studies, and finally go for in vivo studies and enter into the clinical trials. In clinical trials, you have the preclinical studies. In preclinical studies, where your drugs gets approved for testing in humans. So in clinical trials, you have phase 1, phase 2, phase 3, and phase 4. And these three phases, phase 1 will include 20 to 80 participants. And phase 2, this will be a small group, and this will be a large group. And this will be large group. Also, the patients will be included. 100 to 300 small groups of patients will be included. And phase 3 will be large groups of patients or the participants will be included. And finally, FDA will review your applications when you are submitting it for approval. And after approval of from the FDA, it will be taken for phase 4, where the approved drug will be monitored by pharmacovigilance in order to study the effectiveness and the safety of the particular drug, even after being sent it for sale. After it is being given for sale, also it will be entering into a state of approval whether it is having any adverse side effects. So this is the overall summary for this particular how a drug is being decided, how it is taken for development, and finally how it is being how the mechanism of action of the drugs can be evaluated. Thank you.
Thank you, ma'am. Dear participants, if you have any queries, please ask to ma'am.
Am I audible to all? Because nobody is.
Yes, ma'am, you are audible, ma'am.
Is there any questions from the participants? Most of them are silent, I guess. Was the topic useful for you all?
Yes, ma'am, it was quite useful to us.
Okay. Okay, sir. Thank you. So
I
there
want
was a
to
one
ask one
question.
question, ma'am. I have one query, and I want to ask from Shafreen, ma'am.
Yes, ma'am. Yes, ma'am. Sure.
Ma'am, what is the future of the personalized medicine in
Ma'am,
coming
ma'am,
years?
I'm not audible with your voice, ma'am.
Is it audible now, ma'am?
Yes, ma'am. It's audible.
Okay. Ma'am, first of all, it was a great lecture, quite innovative and quite knowledgeable, and I think it will be a good professing in the future. Ma'am, I want to know about the future of the personalized medicine in coming era.
Rekha, can you help me? My ma'am voice is not audible to me.
Yes, ma'am.
What was the question, ma'am?
Ma'am, I want to repeat my question. What is the future of personalized medicine in the coming era?
Uh, Future of personalized medicine. Maybe it was there for the quite long time, but I don't think for the future it will be very useful, ma'am.
Hmm.
Because we
Hmm.
are entering into different, we are encountering into different types of diseases, and
Mhm.
going for a personalized medicine could not be affordable for an. It depends on the affordability of an patient, right? So
Yes, ma'am.
affordability is, it could not be for all the people. So for all the people, even for the layman, I don't think it will be affordable. So, but we are going to encounter so many types of diseases. So, in order to overcome such kind of diseases, I don't think this will be an personalized medicine is a well option.
Okay, ma'am. Okay. Thank you so much, ma'am. Thank you so much
Thank you,
for
ma'am.
such
Thank
a
you.
informative session. Thank you, ma'am.
Thank you. Thank you. And there is one question in the post. How can we extract a particular compound from a plant source? So it depends on the type of the compound you want to extract. There are different methods of extraction which has been available already. So many people are publishing papers on extraction. So what kind of compound you want to extract exactly? So you have to focus on that. Just taking a either if you don't want such kind of specialized or a particular type of compound, you want to see all the compounds which you could be the target. You just go for a crude extract to see the whether the plant is having any antimicrobial activity or whatever type of activity you want to see. Then after that, you can go for some TLC methods, and you can rule out that particular fraction, and then you can go for the isolation. Or you can, nowadays we have GCMS and LCMS to help you out. So you can directly give the fractions for extraction. In small scale, if you want to perform, you can perform using different based on the polarity of the compound. You can separate the based on the solvents. You can use it and you can separate in the lab. Then you can go for a preliminary screening, and then you can go for LCMS or GCMS, and then you can identify the compound of your choice. But if you already have determined, I want this class of, say for example, I want flavonoids. If you have already decided on that, then you can focus on that particular protocol, which is already available in so many papers. I hope I could communicate to that answer.
Hello, ma'am.
Yes.
Uh, Ma'am, I want to know, as per your data, out of 5,000 drugs, only one drug is accepted.
Yes.
Ma'am, I want to know how many
No.
drugs are, uh, sorry,
How many drugs?
how many drugs are established during last 10 years for different diseases? If such is the difference, only one drug is you can say produced out of 5,000 drugs which has been estimated, means the
Yes,
scientists have been
actually,
working.
yes, actually, what we do is a preliminary screening we do in lab, we have 5,000. Say for example, from a plant, we have
Your
around
voice is not
10,000.
audible, ma'am.
My voice is not audible. Okay.
Now, now it's clear.
From a plant source, we have thousands of compounds. Okay. For example, I'm telling you,
Yes. Yes.
out of the 1,000 compounds, we rule it down to 250 compounds in laboratory. So out of
Okay.
250, we cannot purchase all the compounds, right? It is not affordable for us to do that particular experiment. We go for only the top hit. Top hit is only 5. We have only five compounds which we can purchase from our budget, from our research and development budget. We go for 5, and from that 5, we will see only we can give all the 5 for approval, but the clinical trials, all these are very costly. So that is what they are telling, and sometimes it can also lead to the failure. In clinical trials, they may not work properly. Maybe they cannot scale up the product, they cannot scale up the drug. The drug sometimes, when you are going for a large scale production, they cannot have the fine structure which we expected in small scale. That is also happening. So that's
Mhm.
why from 1,000 or 10,000, it is getting cut down to 1 because of the cost, because of the production issues, because of the labor intensive work. So that's why it is getting cut down to 1. So when it is coming for 1 also, sometimes after the drug is being designed and it is taken for testing in the patients or in a large scale of screening, it can also fail. There are such similar cases which are being noted down. Like it has entered into an approval of FDA, but when it is going for a large scale monitoring, it has failed.
Mhm.
So this has also happened in around in 10 years. I think totally 10 in a recent data from Nature. I think they have given only 10 drugs which have they have clearly mentioned. Maybe I can share you. Only 10 drugs are being taken, and in that also only one has been reported out. So this is the situation currently due to financial issues and ethical clearance and so many issues. It is getting failed
This data is from India, ma'am, or from all over the world?
all over the world.
Only 10 drugs.
Yeah.
Okay. And patents, ma'am?
Patents are so many patents. I like to say that there are so many patents being filed, and patents are being listed like thousands and thousands of patents are coming in a year.
Okay. Thank you, ma'am.
Thank you.
Hello, ma'am.
Yes.
Ma'am, actually, before getting into the clinical trials,
Mm.
in vitro, like in vivo studies with animal models will be carried out.
Mm.
Right? That is to check the efficacy and also the toxicological effect of the drug.
Yes, ma'am.
Should it show nil toxicology to enter into clinical studies, or even a meager toxic drug can be taken up to clinical trials?
Ma'am, there is one fixed ratio or proportion for that. I think, ma'am. When you are
Yeah.
when you are filing it for animal model itself, you will be proposing this particular is having this much toxicity in cell lines. Already, I hope you will be proposing that in front of the panel. At that time itself, they'll be telling you this the toxicity level monitors and all they will be giving you. I think so. So based there can be some level of toxicity, but there will be some monitoring levels.
Okay, that is because why I am asking this question is many a times the toxic effect or the death of any animal models need not be due to only the drug.
Yes.
It may be due to individual
Response.
conditions,
Response,
right?
response.
Yeah. So response. So that is why I just posed this question.
Yes, ma'am.
So even if it is having some toxic effect, also it can be taken only if it is up to that level, right? That is
Yes,
your
ma'am. Right.
answer.
Right.
Yeah. Okay. Thank you so much.
Thank you, ma'am. One person has asked the question, what is network pharmacology to drug design? So network pharmacology, like we have one drug, one disease concept, right? Now it is like when we take a drug, we enter into so many responses in our human system. So for that, network pharmacology is playing a very good role, and we use one drug so that all the drugs, all the proteins which are involved in this regulatory response can be identified. So when it is binding to one particular target, what are all the other effects? So that can be studied. So it is a very good technique which has been recently used by so many people, that network pharmacology is helping in drug designing process also. Every person's genome sequence different from each other. So how can we develop a general drug for an individual of sample size, and there is a chance or for an one drug is suitable to one person, which is not suitable to other because of their. Yes, this is what you call it as personalized medicine. So personalized medicine will come into the point here. So which can be monitored continuously. The patient can be monitored, and what kind of drug he can respond. The profiling can be taken, and after the profiling is taken, their genome level, their proteome level, then what are all the biomarkers produced, all these things will be considered, and those person can be given a personalized medicine. But this is what I think some first question was by some ma'am. I addressed for that. It is not affordable for all the people. It is affordable for only few sample size of the few people who can go into this. So this has, if this is going to be happening, then everyone will get a suitable drug for their particular disease.
Hello, ma'am. I have one more question.
Yes, ma'am.
Actually, this network pharmacology and molecular networking, are they same or different?
Molecular networking is different, ma'am. Network pharmacology is you are going to use a drug moiety and you are going to screen against different proteins.
Hmm.
Ma'am?
Yeah, hello?
Can you hear me, ma'am?
Yeah, ma'am. Yeah, ma'am.